Okay, so if we start with digestion, in the mouth you have a tube, the buccal cavity goes to the esophagus, so this is your esophagus. In the mouth you have salivary amylase, and that works at a pH of approximately 7, and that digests or hydrolyzes starch into maltose. So that's called amylase. And basically you're taking amylose, which again you need to remember your structure of your biological molecules. So this is a good type of question that helps you to recap your biochemical knowledge of your biological molecules. So for example, starch is made up of lots and lots of glucose molecules joined together by 1,6 glycosidic bonds. So it's the glycosidic bonds that you are breaking with the amylase. The amylase only works in the mouth at pH 7. When it reaches the stomach, the pH that goes down to pH 2. So the enzyme amylase will denature. In the stomach there are other enzymes. There are enzymes like pepsin, which is an endopeptidase. And basically endopeptidases take long polypeptide chains and they hydrolyze the bond in the middle of a long polypeptide, which is the peptide bond. And that makes shorter chains. That makes them great if you want to work on them with exopeptidases, because the exopeptidases can only work on ends to release amino acids, which is the monomer of polypeptides, or dipeptides. When we come out of the stomach, the pH is very acidic, so we need to have an input which is from the bile in the gallbladder. The bile is actually made in the liver, which sits behind here, and then it's stored in the gallbladder. The gallbladder releases the bile and that neutralizes the acid. So then it's ready to release enzymes that work at a more neutral pH. For example, pancreatic amylase, so this is the pancreas, and pancreatic amylase is released so that any starch that is still in the food at this point can get digested at this point here. We also have pancreatic lipase and pancreatic dipeptidase, and you also have some other protease enzymes that act like endopeptidases and exopeptidases to break down your proteins further. So now we're into the duodenum. This is where we're completing our digestion, so we can start to think about maltose being hydrolyzed into glucose. So that's breaking a glycosidic bond, so we need water to do that because it's hydrolysis. Also in this area you will also have dipeptides into amino acids, and that's breaking a peptide bond. And finally you have your triglycerides. Now the first thing to happen to triglycerides in digestion is that that bile that's in there can emulsify your fats. So first of all you've got emulsification. That is not chemical digestion, that is mechanical digestion. And what we mean by that is you're just taking big drops of fat and making them into smaller ones, which increases the surface area. Once we've increased the surface area, that makes it good for working on with enzymes. The bigger the surface area, the better, the more efficient the enzymes will work, they'll break it down faster. So we have lipase enzyme, which will take triglycerides. Now this is the different one. When you studied your biochemical molecules, you'll have looked at starch being broken down into maltose and then into glucose. That's full hydrolysis down to the monomer. With dipeptides, you're breaking those down into their monomer, which is amino acids. With triglycerides, when you learned this in your biochemical booklet, you learned that triglycerides get broken down into fatty acids and glycerol. In this case, we're not going to break them down into fatty acids and glycerol, we're going to break them down into fatty acids and monoglycerides. So fatty acids and monoglycerides are the products of the lipase hydrolysis of triglycerides. Once you've got those, they then come together to form what's called a micelle. The micelle then travels to the ileum, And it's at the ileum that we're going to now start thinking about absorption. So if we move now onto the ileum, and we look at the ileum at a cellular level. So at the cellular level, you have cells that have microvilli, not to be confused with villi, which are the foldings of the intestine. So if you look at the intestine as a tube, so from the outside it just looks like a cylinder. If you looked inside that, what you would see is the cross section that looks like this. And these are all the folds within the inside of the lumen. So your food is in the middle, and these are the folds, and then within these folds you have villi. Within each villus there is a capillary bed and also a lacteal. So the capillary bed runs really, really close, and then if you look in detail you can see that the cells that line this intestinal wall are just one cell wide. So between the lumen, which contains your food, and the blood vessel, which is going to take up all your nutrients that you've absorbed from your food, there is just one cell to get across. And that cell contains microvilli. That helps to increase the surface area for absorption. So there's 3 different things that you've got to absorb. We've got to absorb amino acids. So amino acids are going to go through a co-transporter protein that will also carry sodium ions. So the amino acids bind along with the sodium ions, and that causes the protein to change shape and push the sodium and the amino acids into the cell by facilitated diffusion. So facilitated diffusion allows amino acids and sodium ions to come in. Now it would quite easily go in the reverse direction with this co-transporter, but you get rid of that sodium really quickly using active transport. So that requires ATP, and that maintains a concentration gradient for sodium into the cell, which means if there is no sodium inside the cell, the amino acids cannot get back out through that co-transporter. They can only travel in one direction. For every different shape of amino acid, you will need a different protein, because each protein shape is complementary to the different shape of the amino acid. The amino acids can then diffuse by facilitated diffusion through a carrier protein out and into the bloodstream at the other side, because there is a concentration gradient for amino acids inside the cell where there's lots of them accumulating to go out into the bloodstream. Similarly, the same kind of thing happens with your glucose. It uses a similar kind of mechanism. Again, glucose binds to the co-transport protein alongside sodium. Because there's no sodium, because you've actively transported it out of the cell, that means that glucose can accumulate inside. It will not go back into the lumen. So then if there's high enough concentrations, it will travel out again by facilitated diffusion, down its concentration and into the bloodstream. Now the difference here, we needed a transport protein because if you remember the properties of amino acids and glucose are that they cannot get through the phospholipid bilayer. But if we're talking about fatty acids and monoglycerides, that's slightly different. So what will happen is the micelle will arrive at the cell, it will dissociate back into its monoglycerides and fatty acids. They will then diffuse through the phospholipid bilayer between the phospholipids. They will then be processed by the smooth endoplasmic reticulum, which will put them back together to make triglycerides. So you make these fatty acids and monoglycerides back into triglycerides. They will then go to the Golgi, where they will have cholesterol added and lipoproteins, and that will make a chylomicron. The chylomicron will then be moved out into the lacteal, not the bloodstream. The bloodstream is full of plasma, full of water, so we exocytose, so they go out in a vesicle. So this is your chylomicrons going out to the lacteal by exocytosis. And that is absorption of your fatty acids and glycerol, or fatty acids and monoglycerides, your absorption of your amino acids, and your absorption of your glucose. Note that all 3 of those mechanisms will need a source of ATP, because exocytosis also requires ATP, just like active transport did for the sodium.